And you've got your tetrads, and you've got your packets for Sarcini ferments. How do you classify bacteria? You can look at what they look like in the microscope. You can look at the colony appearance. How do they grow on a Petri plate? You can put them all through biochemical tests and see what they can do. You can look at their chemical composition and analyze that. You can see how they react in your blood, how you make antibodies against them. If you make antibodies against one type of bacteria, but those antibodies don't work against this other bacteria, you can say, oh gosh, these must be two different types of bacteria. So you can identify them that way. And the sixth way is to actually just get in there and look at their DNA. How many G's and C's do they have, the actual DNA sequence, when you find out the order of all 4 million letters, or maybe half of that or whatever. Do that. We aren't really going to get into this too much. We're pretty much going to characterize the bacteria by what diseases they cause. Here's a figure that shows some relative sizes. Sizes are really important to me. Here's a human hair. It's magnified a lot. And then here's a little bunch of things. There's ragweed pollen, a white blood cell, a red blood cell, a yeast cell, and then some little teeny weeny bacteria. Notice how the prokaryotic cells are really small? Okay. If you look inside that, then here's your E. coli, about 2 millimeters long, got a little bit of a curve on it. Here's your staphylococcus, and even tinier than that, way tinier, down in scale, you get the Ebola virus and the rhinovirus. Rhinovirus, do you know what that causes? Oh, there. Cold in your nose. Domain typhoid. I put this slide up only so that you'll remember the order of these, right? And you remember the same? Dumb King Philip, or David, either one of those two guys, calls our family gatherings silly. Or, yeah, dopey kids playing catch on freeways get squashed. I'm told that if you're ever trying to make up a password for a computer, don't use your social security number, don't use 1, 2, 3, 4, 5, 6, don't use QWERTY. Take a sentence, maybe the first sentence of your favorite book, and use the first letters of those words. So, it was a dark and stormy night. Yeah. Okay. A species, this gets a little bit murky for bacteria because they swap around. They don't breed true, as we say. They are, in fact, promiscuous. But you can call it a species, or be generally agreed on as a species, if the traits and the qualities that were on the previous slide, the 6 things, are pretty much the same, and they're different from other bacteria who are quite different. Within a species, you can have kind of a subspecies, a strain or variety. In E. coli, you have a lot of E. coli in your stomachs right now. You probably don't have that strain 0157H7. That's the one that the jack-in-the-box bacteria. Okay? There's also perhaps a type. This is might be different from other features in the strain by 1 or 2 genes. Okay, so there's ways of defining more and more what these bacteria are all about. We've already been through this. If it's a capital B, it's a genus, and you already know a few things about the genus of Bacillus. It can make spores. Not all rod-shaped bacteria can make spores, but capital B Bacillus, yep, it can. Mostly these are bacteria that need air to live. Clostridiums, that genus, mostly are bacteria that don't like air, that don't live in air. So that will be maybe a 1 or 2 point question on exam. Once again, Staphylococcus and Streptococcus, capital S, it's the genus. Little s, oh, okay, it's little round cells in bunches, whatever they are. Okay, so any questions? You can go over those.
Are we doing this today? Or?
Yes.
Okay.
Yes, because,
Did we need schedule?
no, you don't. Oh, no, not this.